Hello and welcome to the virtual lecture on homologous chromosomes, the sexual life cycle, and meiosis. Our first topic, homologous chromosomes, is really a continuation of chromosome structure and chromosome numbers, which we discussed in a previous module when we were talking about mitosis. So homologous chromosomes are pairs. As humans, we have 23 pairs of homologous chromosomes. So, for example, humans have 23 different types of chromosomes. There's number 1, 2, 3, all the way to 22, and then finally the sex chromosomes, X and Y. Now, if you were to look at just one of those types of chromosomes, for instance, chromosome number 1, within each of your somatic cells, you have 2 copies of chromosome number 1, as well as for 2, 3, and so on. Why do you have 2 copies? Well, you inherited one copy from your mom and the other from your dad. Together, the maternal version of chromosome 1 and the paternal version of chromosome 1 represent a single homologous pair. Now again, we have 23 pairs just like this. So we're going to take a moment to describe the ways that the 2 chromosomes within a homologous pair are similar and also how they're different. So I'm going to make up an example here. I'm going to say we're talking about human chromosome number 2. We all have it. It's the second longest of our chromosomes, contains many genes. And we're going to start with how the 2 chromosomes within a homologous pair are similar. And you can already see 2 of the similarities just by looking at the 2 chromosomes that I've drawn. One thing you might notice is that they're the same size, or the same length. And that is true, so we're going to write that down. Now, when we say length, remember a chromosome is a really long DNA molecule, so they roughly have the same number of nucleotides. So just to give you a real example, if we were talking about human chromosome number 2, that chromosome, which we all have as humans, is 242 million nucleotides long. So what I'm trying to say is both of these are 242 million nucleotides long. That makes sense. They're the same type of chromosome. How else are they similar? Remember this portion of the chromosome? It's kind of like the waist of the chromosome. It's called the centromere. Notice how their centromeres are at the same position. So the waist is at the same spot. So that's always true for the 2 chromosomes within a pair. So we're going to say same centromere position. And there's one more way that they're similar. You can't see it, but it's the most important of the 3. And that is that these chromosomes within the pair both contain the same exact genes. Not only that, but those same genes are located at the same positions along the chromosome. So I'm using a term here called locus, loci, plural. I haven't defined it. I'll define it in a moment in the PowerPoint slides, but it basically just means the specific location of a gene along a chromosome. So all I'm saying is these two chromosomes have the same exact genes that are intended to code for the same exact proteins, and those genes are located exactly the same spots on both chromosomes. So keeping with my kind of a real world example here, I'm going to pick a real human gene that is located on chromosome number 2. We all have it. The gene that I'm choosing is called the lactase gene. We're going to call it LCT for short. It's not the gene that matters. I could pick any gene. It's the fact that since these two chromosomes are within a homologous pair, they both have the LCT gene that is intended to code for the enzyme called lactase, which allows mammals and other organisms to digest and break down milk sugar, lactose. So you could look at any of the hundreds of genes that are located on these chromosomes, they would all be at the same spot. So it sure sounds like they're identical. So how do they differ? How do they potentially differ? That's also very important. Now, the way that they potentially differ is the specific versions of the genes that they have. So remember, we learned that for any given gene, there's often different varieties, which we call alleles, that exist within the population. So they could potentially have different alleles. Now again, it's important to note that they do have the same genes, but they could have different versions of those genes. So keeping with this example, if this is the lactase gene, well, maybe from the dad, they got the lactose intolerant allele. So that's an allele that turns off this gene past age 7, so they can't digest lactose. Maybe from the other parent, from the mom, they have the lactose tolerant gene that allows them to continue to express this gene their entire life. So same gene, intended to code for the same protein, different versions. Now, the other reason that I'm choosing to cover homologous chromosomes in depth now and not earlier when we were talking about, things like sister chromatids and kinetochores and centromeres, the reason is that it connects with meiosis and sexual reproduction. Because as we're going to learn, the whole goal of meiosis, which is a form of cell division, is to separate each of our 23 homologous pairs and to give one of these two chromosomes to your child. So regardless of whether we're talking about males or females, it could be sperm or eggs, but when they produce sperm or eggs, which are called gametes, they're going to give that gamete one of these two chromosomes. Now, they can't give them both, And as we're going to learn, it's like a coin toss. It's a 50-50 chance as to whether they're going to give their kid the one from their mom or the one from their dad. So that's why it's relevant to cover this now. All right, so as I said, this term locus just means the location of a specific gene upon a chromosome. So spread out across our 46 chromosomes in our cells, we have about 20,000 different genes that each code for a unique protein. So we can say as humans, we have about 20,000 different gene locuses, or 20,000 different gene locations. Only organisms that are of the same species have the same number of homologous chromosomes and the same genes at the same spot. That's what makes them look like a human, or look like a dog, or look like a cat, and function like a cat, dog, or a human. So for example, dogs, they have 39 homologous pairs, and they have genes that make them look and function like dogs across those chromosomes. So it's specific to a species. You've seen this picture before, but it's especially nice to cover it now because in this karyotype, they've organized and grouped together all 23 homologous pairs. So it's important to note that if you were to look inside the nucleus of one of your cells, your homologous chromosomes aren't just nicely lined up like this and next to one another. They're actually not next to one another, except when a cell is undergoing meiosis, as we're going to talk about in a bit. So it was a computer program that put them and lined them up like this, but you can really see how similar they are. So here's the actual chromosome number 2, same length, their centromeres the same spot, they contain the same genes. Same thing for any of the other pairs. If you look at, for instance, chromosome 18, same length, centromeres the same spot. And I want to point out that the way that we name or number chromosomes is in terms of their descending length. So chromosome number 1 is the longest, has the most genes. Chromosome 22 is the shortest with the least amount of genes. Then here we have a special, unique pair of chromosomes. These are referred to as the sex chromosomes. We'll talk about those in a moment. Oh, I guess here's the moment. The 23rd pair are called the sex chromosomes. Biological females have 2 X chromosomes, shown here. Oops. A biological male has 1 X chromosome and also a little Y chromosome. So these largely, I shouldn't say these, it is the Y chromosome that largely determines male biological development, sex determination. So kind of like the default is to develop into a female, but if you have the Y chromosome, it will change some things around to have you develop as a male. The X chromosome is kind of like any other of the 22 chromosomes. It has a ton of different genes, most of them unrelated to sexual development at all. So for instance, genes that code for blood clotting proteins, genes that code for proteins that sense light in your eyes, all different types of genes. It's really that Y gene that's involved in sexual development. But what you need to know is that the 23rd pair, those are called the sex chromosomes, XX for a female, XY for a male. Now, I want to say one more thing on this slide. Up here, if we're not referring to the sex chromosomes, all other pairs, the other 22 pairs from here to here, those are called autosomal chromosomes. So chromosomes 1 through 22 are called autosomal. The last pair are called sex chromosomes. Moving on, the remainder of this video is going to be about the sexual life cycle of animals. We're going to place greater emphasis on this part of the life cycle, where we do a very special type of cell division in preparation for sexual reproduction called meiosis. But I will briefly talk about other parts of the cycle as well. So we're looking to understand, how does a parent, any type of animal, doesn't have to be a human, how does a parent transmit or pass on their genes to their child? Physically, how do they get their genes ready? And then how do they give them to their child? How does that work? The first step in this process is meiosis. And it's a preparatory step for sexual reproduction. And the goal is to produce a unique type of cell called a gamete. Male gametes are called sperm, female gametes are called eggs. And as we're going to learn, these are very different from the other cells in our body, somatic cells. And we will make sure to highlight those differences. So that's the goal, to produce these cells. Now this is a very important slide because it really highlights all the main points about meiosis. And again, that's the emphasis of this video. So why do we do it? Very simple. In preparation for sexual reproduction. Who does it? Or what? Pretty much all animals, most plants. I say most because a lot of plants can also do budding or asexual reproduction as well. But also, the vast majority of plants reproduce sexually and actually prefer to. And also many funguses like mushrooms, for instance, and molds. Third, what are we looking to achieve as we do this process? What's the goal? Really important. First goal, we want our daughter cells to be unique. We want the sperm and egg to be what we call genetically unique. Each one to have different random combinations of alleles. That's very different from mitosis, because in mitosis, we wanted the daughter cells to be identical to the parent cell. That is not the case here at all. So that's a good thing. We want to shuffle up our genes. We want to shuffle up the alleles and produce different combinations. Number 2, we want to cut things in half. Not physically, but in terms of numbers. So we want to take the number of chromosome sets and the total number of chromosomes and to cut both those numbers in half. Very important. It's important because, well, think of it this way. If the cells in your body have 46 chromosomes, and let's say you did mitosis to produce sperm or eggs, so your gamete, your sperm or eggs, had 46 chromosomes. And same thing from the female. She produced a gamete that had 46 chromosomes. And then they fertilized. That would not work out. The child would have 92 chromosomes. Big problem. I don't even think that would work. So we don't want to just keep doubling the number of chromosomes each generation. We want to keep it consistent. Humans, as a species, have 46 chromosomes. So to achieve that, we have to, like I said, cut the number of chromosomes and the number of chromosome sets in half. so that when the sperm and egg combine, each one will donate 23, and together, the new child, the fertilized egg, will have 46. Okay? So, cutting things in half. First thing we're going to cut in half is the number of chromosome sets. Most of your cells, including the parent cells called germline cells that we start with in meiosis, they have 2 sets of chromosomes. They're considered to be diploid. 1 set from your mom, 1 set from your dad. When we're done with meiosis, we want the resulting daughter cells, the gametes, to only have 1 set of chromosomes, meaning they're haploid. So we're going to go from 2 sets to 1 set. And then lastly, most cells in your body, including the cells that we're going to start with, the parent cell in meiosis, have 46 chromosomes in total. But as a result of meiosis, we're going to cut that number in half. And the resulting gamete cells will have 23 chromosomes. So in summary, we're looking to go from 2 sets, diploid, to 1 set, haploid. And we're looking to go from 46 chromosomes to 23 chromosomes. And we want them to be really genetically unique. So if you can keep that in mind, most of the mechanics of the process should make sense. So that's just my introduction to meiosis. We're going to continue along here in the sexual life cycle. The next step, actually not the next step, I guess here I'm just talking about gametes. Nothing new here. Human gametes, they're cells, but they're very special because they only have 1 set of chromosomes. They're haploid, and they only have 23 chromosomes. We already covered their names. Moving on, when 2 animals mate, potentially their 2 gametes will fuse. During a process called fertilization, sperm fuses with the nucleus of the egg, and they basically combine their chromosomes. And fertilization, like the numbers of it, it's really easy to understand because it's basically the opposite of meiosis. We're going to take everything that we just cut in half and just double them back to what they were. So during fertilization, we're going to go from haploid to diploid. We're going to go from 23 chromosomes to 46, if we're talking about humans. And we have a name for this cell that is the result of sperm and egg fused together. It is called the fertilized egg, also known as a zygote. We all started like this. All animals start as a single cell. Pretty wild. And then through many rounds of mitosis, and you have certain cells becoming certain tissues, a lot of growth and development, until finally you're an adult. Several trillion cells. And that's what I just talked about here. Going from a fertilized egg to an adult, and that is strictly mitosis. So that sums up the cycle for the remainder of this video. Again, we're going to focus right here on this portion of it, meiosis. First off, where does it take place? Specifically, it takes place in the reproductive organs. It's at testes in males and ovaries in females. Another important distinction about meiosis, especially relative to mitosis, is that meiosis involves 2 rounds of cell division. So Basically, you have a parent cell. Parent cell. It's going to divide. I need more room than that. Let's see. You have parent cell. It's going to give rise to 2 daughter cells. That's 1 round. Then they're going to divide again, each of these cells. Man, this pen is really hard to use. And in the end, you're going to have 4 daughter cells, which we call gametes. So that's what I mean by 2 consecutive rounds of division. The first round is called Meiosis 1 with a Roman numeral. The second round is called Meiosis 2. We're pretty much only going to talk about Meiosis 1, and that is because everything that we're looking to achieve in meiosis, cutting the chromosome numbers in half, the sets in half, is going to be achieved during Meiosis 1. And again, the outcome is going to be 4 genetically unique daughter cells. Now, this is a great picture right here because it summarizes most of what you need to know about meiosis. Now, what we're looking to do is we're looking to take each homologous pair. Now, what's nice here in this picture is they only focus on 1 pair. It would be really messy to look at 23 pairs, you know? And we're going to take each pair, and it's basically like a coin toss. Let's say heads is blue, tails is red. And we're going to randomly select one of these 2 chromosomes and give 1 to each gamete. And in doing so, the 2 resulting cells will have 1 set of chromosomes. And we're going to cut the number in half. That's pretty much all we're looking to do here. So, in summary, meiosis is the random separation of homologous pairs. Now, if we're talking humans, you just have this same process going on 22 other times because you have 22 other pairs. Each one is a separate coin toss. And as you can kind of imagine, there's a lot of different possibilities. A lot of randomness. So, next, we're going to go through my drawings of meiosis. Now, I want to point out that meiosis is similar to mitosis in terms of the mechanics of it, in terms of the names of the stages. The key difference is how we align our chromosomes during metaphase. But other than that, pretty much everything you learned about in mitosis is the same. So I'm not going to test you on that. I'm not going to test you on microtubule filaments again, or the centrosome. Okay? You just need to focus on 2 things. How the chromosomes are aligned during metaphase, and then also another important event that happens during prophase. But there's not going to be a whole bunch of quiz questions with pictures of my drawings. There's only going to be one, and I'll tell you this, it's a picture of metaphase. So make sure to pay attention to that. But it's an impossible subject to teach unless you actually draw it out. So, just as I did when we covered mitosis, we're going to have a simplified cell. Instead of it having 46 chromosomes, it only has 4. So it's actually the same cell that we used when we covered mitosis. So it's a 2n equals 4. This here in our picture, this is the original parent cell. So the cells in the ovaries and testes that are eventually going to give rise to gametes, the original parent cells, are called germline cells. And germline cells are really no different than any other somatic cell. They're diploid, they have 46 chromosomes, 2 sets. So, looking at our example here, let's fill in these blanks. How many chromosomes do we have in total? Well, we can count them. 1, 2, 3, 4. Secondly, how many sets of chromosomes are there? There are 2 sets. We have a paternal set and a maternal set. So we have another term for that, which we've already learned about in the course. This is a diploid cell, 2 sets. Now again, we're keeping it simple. We're going to say there's only 2 types of chromosomes. There's only 2 homologous pairs. We have chromosome number 1, which is going to be our big chromosome. You have 1 from the mom, 1 from the dad. And we have chromosome number 2, the little chromosome, 1 from the mom, 1 from the dad. Now keep in mind, what are we looking to do? We're looking to generate daughter cells that have half of these 2 numbers. So we want the daughter cells to have 2 chromosomes in total and only 1 set. So I'm going to keep jumping back and forth between the slides and my drawings. Nothing new here, I've already said this. And now we're ready for prophase of meiosis 1. This is one of the 2 phases that you want to pay attention to. Now I've learned over the course of a year of making these videos that it's better to have my drawings laid out beforehand than to waste time drawing them out during the video. So as you can see, I've already got things ready here. So here's prophase 1. Just like prophase of mitosis, the nuclear envelope breaks down. We start to build our mitotic spindle. And we start to grab and attach to chromosomes. But what is different is that during prophase 1, the chromosomes within a homologous pair become physically attached to one another. Now I can't draw this because I don't think anyone could in 3D, okay? But they're actually in reality superimposed on one another. So one's on top, one's on bottom. So it's like 2 pieces of paper laid on top of one another. We have a name for that when homologous chromosomes become physically attached. We call each pair, it's kind of a convenient term, sounds like what it means. That's a T. We call each pair a tetrad, like tetra for 4. So this is 1 tetrad, here's the other. Now if we were talking about humans, you would have 23 tetrads, just like this. Now never, at any other point in your life, in any of your cells, do homologous chromosomes physically associate with one another. They're usually doing their own thing in different parts of the nucleus. This is the only time that the 2 chromosomes come together. Now, something happens when they come together, and it's an event that greatly shuffles up our genes, our alleles, at random. What happens is, it's really kind of crazy that it works, because it sounds dangerous, but this happens in all species that are undergoing meiosis. And what happens is, the two homologous chromosomes actually swap DNA. They actually break off a little piece of their chromosomes and then they trade pieces. Now, what's crazy is that they can break the chromosome at any point, and that's the randomness of it. They could break it here and only trade a little sliver. They could break it here and trade a big chunk. So let's pretend they broke it right here. The result would be something like this. They actually, trade alleles. Now each pair is going to do that. They can do it multiple times actually. So let's say it also happens down here. Now actually I'm going to show it on the bottom just to highlight it. It can happen anywhere. But it does have to happen at least once per tetrad. So that's really cool. Now, the result is that you have what was once a completely maternal chromosome. These used to be all alleles from your mom. Now it's like 95% of your mom's DNA, but 5% of your dad's DNA. You as the individual undergoing meiosis. So you get all these different combinations of genes. And that makes sense because, as we said, a goal of meiosis is to randomly shuffle our alleles. And that really helps us do that. So I'm not going to draw these little shadings from now on just because it makes it messier and harder to keep track of maternal versus paternal, but just do realize that it does happen. And now I've said all this, but I haven't given a name for the process. The process of exchanging alleles between the two chromosomes within a homologous pair is called crossing over. Now let's take a look at what we have to say about this in our slides. Now, as usual, I'm only going to highlight things that I haven't already said. There is nothing new to say on this slide, so we're going to move on. Here is just a picture to show you how the process works. Again, they break off pieces of their chromosome and trade them and swap them and then reattach them. All you need to know from this slide is that, again, this crossing over and swapping of DNA shuffles genetic variation. It shuffles alleles. Very important. Now we're ready for metaphase of meiosis 1. And this is that other phase that's important. It's the most important one. So moving forward, here we go. Now, this should look a little bit different than metaphase of mitosis. I actually have a picture of mitosis, which is nice, just to compare. Remember, in mitosis, metaphase would look like this. You would take every individual chromosome and then pull apart the sister chromatids. But as you can see, that is not the case in meiosis. Very different. And it's this difference that's going to allow us to achieve two of our goals: cutting the total chromosome number in half, and cutting the number of sets in half. And it's very important for you to be able to describe what you're looking at here. And what you're looking at is, we have homologous pairs lined up at the equator of the cell. And then during anaphase, we're going to randomly pull apart these homologous pairs. Now, this is really that coin toss that I was referring to. The gamete could get this one, or it could get this one, but both can't happen at the same time. It could, but that would be a major problem. We'll talk about that later on. So, I want to show you another picture. Since this can go either way, you could have blue on the top or blue on the bottom, or either way here, it's a 50-50 chance. Let's show an alternate scenario. By chance, it could also end up looking like this. So, in my simplified example, you can actually calculate the number of gametes that are unique that you can produce. And it's basically 2 to the n power, where n is the number of homologous pairs. So in this example, it's 2 squared. We can make 4 different types of gametes. Now I'm saying that because for humans, you can get so many more combinations because we have 23 pairs. So using that same formula for humans, we have 23 homologous pairs. There's 8.3 million different ways that we can line up and subsequently separate the 23 pairs. That's crazy. And you can think about it, so let's say, you're the individual that's having kids. The pink represents your mom, the blue represents your dad. By random chance, you could produce a gamete that has way more chromosomes from your mom than from your dad. Hypothetically speaking, it would be like getting heads 23 times in a row, but it could happen. You could produce an egg or sperm that only has chromosomes from one parent, so that your child gets no genes from one of their grandparents. But usually it's somewhere down the middle on average. But just the random chance of it is interesting. So back to our scenario 1. We have a name for this event of homologous chromosomes being lined up at the equator. Lined up randomly. And the name for this random alignment of homologous chromosomes is called independent assortment. I also want to say one more thing. It's a 50% chance as to whether the gamete will get the maternal or the paternal chromosome. Either could happen. Now we've finished the 2 important phases, so we're actually going to skip past anaphase, past telophase, and just skip to the end result. So how would that look? The 2 daughter cells would look just like this. Now, let's tally the total number of chromosomes in each cell and also the number of chromosome sets in each cell. Starting with the total number of chromosomes, just looking at one cell. How many do we have? Well, we have 1, 2. How many sets does each cell have? Well, just one set. So if you're to look at chromosome number 1, the big one, now they only have 1 copy of it. They got the version from the dad. If you were to look at the little chromosome, chromosome number 2, they only have 1 copy of it. Happens to be from the mom. So there's only 1 set of chromosomes. And we have a name for it. Since it has 1 set, It is a haploid cell. So we achieved all the goals we were setting out to reach. Why do we have to go through a whole nother round of cell division? Isn't this it? No. The answer is you have to do a whole nother round of cell division just for one simple reason. And that is, the chromosomes that we currently have are in their duplicated form. And you could not give your newborn child chromosomes that are already duplicated. What is the child supposed to do with that? The fertilized egg, the child, the offspring, has to go through its own first cell cycle, including S phase. That would be messy. If it, comes to S phase and it already has its chromosomes duplicated, what is it supposed to do? So what I'm trying to say is you have to give your child chromosomes in their unduplicated form. So again, we're not going to focus on it, but it looks like this. It's actually just like mitosis. We're going to pull apart sister chromatids so that we can get 4 daughter cells that look like this. Now remember I said in our hypothetical cell here, there's 4 different types of gametes that we can produce. Here's one of the types. They can have a big blue and a little red, or it could have turned out this way. We could have got a little blue and a big red. Or if you really want to get into it and look at scenario number 2, it could have turned out this way. You could have got big red, little red, or big blue, little blue. All 4 of these outcomes are just as likely to happen. So, I think that is it for the drawings. We're going to go back here. It's important to note that this process of independent assortment, just like crossing over, it greatly contributes to the shuffling of genes. Because there's so many different combinations, so many different possibilities. This is a nice diagram that takes you through everything that I just drew out in my drawings. Same example, a cell, a hypothetical cell with 4 chromosomes. Red is from the mom, blue is from the dad. And it shows you exactly everything that I showed you, but it's nice because it's all in one picture. So, just to say it again, when you randomly pull apart your homologous pairs, again, it's a 50% chance. It's like a coin toss as to whether you get the one from the mom or the one from the dad. Now, I don't like the pictures from the book. They look messy because they show the crossing over, so it's really hard to understand what you're looking at. So you can kind of ignore these for now. I just want to emphasize this statement right here. Okay? So, if we're talking about human cells that are undergoing meiosis, the 2 cells that result from the first division, meiosis 1, they would be haploid, and they would each contain 23 chromosomes. So, I thought I had this somewhere. Let's see. Here it is. Let's relate everything that we've learned here in our hypothetical example to actual humans. Okay, so this arrow represents meiosis occurring, Okay, so at the start, we have germline cells in the testes and the ovaries that are diploid, and they have 46 chromosomes. So these are the parent cells. We start with 46 chromosomes. We start with two sets, a maternal set and a paternal set. When we are finished with meiosis, the resulting daughter cells, the 4 of them, which we refer to as gametes, will have 23 chromosomes in total and 1 set. In a very simple sense, we cut these numbers in half. Okay? So definitely know all of these numbers as they relate to humans. Alright, so this whole slide discusses meiosis 2. I already said all I wanted to say about it. And as you can see, there is an asterisk, so it is not a testable material, so I'm not going to waste your time here. We're going to move on. And we're going to finish this video by talking about 2 more things. First, what is the point of all this? I keep saying that it's so great, so beneficial to shuffle your genes and have all these random combinations. Why? Who cares? So we're going to talk about the advantages of the sexual life cycle, the advantages of all of this shuffling. Then lastly, we'll talk a little bit about mistakes that can happen during meiosis. So first, in the sexual life cycle, there are 3 events or 3 processes that contribute to the shuffling and the randomness of the production of offspring. I should have started the whole video by saying this. Many of us have siblings. If you don't, imagine someone that has siblings. If those 2 siblings are not truly identical twins, they can be so different in so many ways. So we're trying to understand why siblings are so different. How do we get such tremendous variation in genetics, which in turn affects their development, their physiology, their height, their hair, eye color, behavior, everything about them. Now I'm going to throw a number out there, and I'll explain how we get to that number in a bit. But I find it completely mind-blowing. It's such a big number that I don't even know, it would probably take up the whole wall to write all the zeros. But if 2 individuals, 2 parents have kids, with the exception of identical twins, they would have to have 4 decillion. Yeah, I just said the word decillion. Again, I don't know how big that is. It's really big. 4 decillion kids to witness or observe all the different types of genetic varieties that those 2 parents can produce. So just try to wrap your head around that. If you have siblings, there's potentially 4 decillion different varieties of yourself, or brothers and sisters versions, That's crazy. So how does that happen? How do we get all this shuffling, all this variation? The answer is just those 3 events that we covered. Number 1, crossing over, which again happens during prophase of meiosis 1. Number 2, independent assortment, the random alignment and subsequent separation of homologous chromosomes during metaphase of meiosis 1. And then lastly, fertilization. Because if each parent is producing many different types of gametes, there's all sorts of ways that those different gametes can combine. So this has an asterisk, but it's interesting, so I'm going to say it. I actually already said it, so I'll be quick. Humans have 23 pairs of homologous chromosomes. Therefore, there's around 8.4 different possibilities that your gametes could look like just in terms of independent assortment alone. Taking into account the randomness of both independent assortment and crossing over makes it so that each individual, each human, can produce 70 trillion different genetically unique types of sperm or egg. 70 trillion. So if you have the mom producing 70 trillion different types of eggs, potentially, and the dad producing 70 trillion different types of sperm, there's all these different ways that those 70 trillions can combine. That's where we get that number, 4 decillion. So you really, truly are unique, not just in terms of our species, but in terms of the different ways you could have turned out, from your two parents. So you still might be wondering, that's great and all, those are big numbers. Wow. In a biological sense, why is that a good thing? And the answer relates to evolution, okay? The more random combinations of traits and alleles that offspring, children have in the population, the more likely the population will be able to adapt to the surrounding environment. Because you never know which particular combination of alleles and traits will be the best to have at a given place and a given time. But the more combinations you have, the more likely if, something harsh comes around in the environment, a predator, a pandemic, something in the weather. Now these are more easy to understand if you're, I guess, an animal, not a human in the wild. But you want to have different varieties because you never know which variety might be the best to have in the wild. So it helps us evolve and it helps us not go extinct. Lastly here, we're going to talk about potential errors during meiosis. What could go wrong? Now, an error during meiosis is known as non-disjunction. And it is the failure of a pair of homologous chromosomes to separate from one another during anaphase of meiosis 1. So let me go back to my drawing. Now the goal here, if things go to as they're supposed to go, the goal is one of these chromosomes to go this way and the other to go this way. But sometimes we make a mistake and we grab the whole chromosome, the whole pair. So something like this can happen. Oops. Let's see if I can do this. I can't. But I think you understand what I'm talking about. What if we reel in the whole pair? That's a big problem. Because this daughter cell up here will be missing that entire chromosome, whereas this daughter cell down here will now have an extra copy. That could potentially be a huge problem. that's what non-disjunction is. It's actually, unfortunately, fairly common. So what are some, I don't like to call them diseases because they're not. There's, as we're learning in this class, there's all different ways that you can have an ailment. I guess that's a general term I can use. It could be a hereditary disease due to you inheriting a broken, mutated allele from an ancestor. It could be cancer, the generation of a broken allele during your lifetime. It could be a disease caused by a virus or a bacterial cell. And this is another issue. These are what I like to call syndromes or conditions that are the result of abnormal numbers of chromosomes. So like missing an entire chromosome or having an extra copy of a chromosome. And it's these abnormalities in chromosome numbers that arise from non-disjunction. So I say they're common, they are. Pretty much most miscarriages that happen, and they happen to everybody, 10 to 25% of human conceptions result in a miscarriage. The reason that that's happening is because non-disjunction happened during meiosis in one or both of the parents. Now, if you have non-disjunction and your child is missing one of our 23 types of chromosomes, there's only one way that the child can live in that case and be viable. And that is if they're missing one copy of the X chromosome. It's a syndrome called Turner syndrome, and it's basically an individual that only has one X chromosome. They're biologically females, but they do have some characteristic symptoms. If your child is missing any other chromosome, it's going to result in a miscarriage. You can't just be missing a whole chromosome. And that should make sense. Each chromosome has hundreds to thousands of essential genes that we need to produce to carry out our vital functions. And if you're just missing a whole chromosome, it's not going to work out. Now, on the other hand, there are many conditions where you can have an extra copy of a chromosome and be completely fine. A few of these you wouldn't even know that you had it unless you were to look and visualize your chromosomes. So these are our last two slides. So first, if you have non-disjunction and you pass on an extra copy of a chromosome to your child, now let's just imagine. Let's pretend this is chromosome number 21. Let's pretend this is the dad. So he had non-disjunction occur. So he's going to give his child 2 copies of chromosome number 21. But it still needs to fuse with the egg. And the egg also has a copy of 21. So once fertilization happens, the result is going to be a fertilized egg with 3 copies of chromosome 21. That has a name that's called Down syndrome, also known as trisomy 21. 3 copies of chromosome 21, as you can see here in this picture. So that's one example of a condition where you can have 3 copies, 47 chromosomes in total. You might be wondering, why is that a big deal to just have 1 more copy? You're not missing anything. It kind of throws off gene expression because you have extra dose of all the genes on chromosome 21, it also throws off mitosis because you have this extra chromosome to line up. Lastly, there are several different conditions where you can have 3 sex chromosomes. 2 of them, you would have no idea that you have it, and they're fairly common. A female can have 3 X chromosomes. They just might be slightly taller, it's the only difference. A male can have 2 Y chromosomes, and you would never know. Another syndrome where a male has an extra X chromosome, that has a name that's called Klinefelter syndrome. And they have more female development characteristics and a possibility to be susceptible to learning disabilities. So I know I'm covering these very quickly, very superficially. It's because it's not testable, but I do want you to understand, in the broad scheme of things, how, Klinefelter and Down syndrome, how those are different than, say, hereditary diseases like cystic fibrosis or sickle cell anemia or a virus like the flu. Okay? Very different. And they are the result of non-disjunction occurring during meiosis. That's it for this video. Thank you for listening.